Hello, in this session we evaluate isomerism. The term isomerism simply means the existence of different structures of the same molecule. An isomer, therefore, is an organic compound which has a distinct structure that differs from another structure that has the same molecular formula. Let's take an example of what we have on the board. We have said that isomerism is the existence of different structures of the same molecule. If you want to represent the different forms of isomer of the molecule of pentane given that is C5H12, we will proceed to represent the structure of the first form, in which there is a continuous chain of 5 carbons. To be able to represent a different structure form, we would break the chain from 5 into 4 and introduce a hanging species. Given that it's not balanced, on the left there are 2 carbons, on the right there is 1 of the hanging species, we can still balance this and create a third structure form of the same molecule. In this case, we will have that kind of arrangement. Now, to complete the displayed structure for these molecules, we represent all the hydrogens present following the carbon rule of each carbon having only 4 bonds around it, and no more and no less. And in this case, our final structure will look like what you can see here. The second species, the same rule will be applied of having 4 bonds around each carbon. And as we do that, comprehensively, we find that the displayed formula shows a marked disparity from the first one. The third one, the sequence continues, carbon rule, 1 carbon, 4 bonds around it. 1 carbon, 4 bonds around it. So as we do this, again we see that the third species has a clear, distinct structure differing from the first 2. Now, separating them, as you can see, we could create the condensed form of this, which is CH3, CH2, 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 and CH3. This could still be condensed further into CH3, CH2 as a recurring molecule, there are 3 of them, into that. So either this or that represent the condensed structure of pentane. Now, the next species condensed will exist as CH3, CH2, CH with a hanging molecule of methyl and CH3. The third species in condensed form is CH3, carbon in between with a complete ring of methyls as displayed there. So then, how do we name these differing organic structures of the same molecule? The first one is simply pentane. The second one, the chain has changed from 5 carbons to 4 carbons. So it's a butane with a methyl in position 2. So we call it 2-methyl butane. The third species has changed from a network of 5 to a chain of 3 with 2 principal methyls on the second carbon. So we could call it 2, 2-dimethyl, 2,2-dimethyl propane. Now 2,2 to refer to the position of the hanging species, di to mean that there are 2 hanging species, and methyl, that the hanging species is a methyl molecule, and propane to mean that there is a chain of 3 carbons in a row. So therefore, we can agree and say that pentane displayed structure, 2-methyl-butane displayed structure, and 2,2-dimethyl-propane are structural isomers of the same molecule known as pentane. Therefore, structural isomers are different structures of the same molecule which differ in respect to the arrangement of carbons in each structure. Structural isomers differ in respect to the arrangement of carbons in each structure. The second example of isomerism would be that of pentene. And pentene has the formula C5H10, which means it's an alkene. And when it's an alkene, it means there is a double bond. How do we represent this? We could begin by saying there are 5 carbons in a chain, and there is a double bond. The position of the double bond will change into 1, 2, 3, 4, 5, from position 1 to position 2. It will again move from that position to the middle, which in this case is on the left-hand side. Now, when you complete the structure, you get H, following carbon rule, that each carbon has only a maximum of 4, no more, no less, bonds around it. So as we represent this, let's see what this comes to. There we have 1, 2, 3, 1, 2, 1, 1 balanced, 1, 2, 3 balanced. There 1, 2, 3, 1, 2, there 1, there 1, there 1, 2, 3. So as we look at this structure, we find clearly that there are 3 principal ways in which you can represent the same molecule of C5H10. However, when you name it, we are going to find that there are only 2 possibilities, in the sense that the first molecule is named from the double bond side, which is 1, 2, 3, 4, 5 carbons, so it is a pent 1 in. The second molecule from the closer side, either left or right, because the bond is after the second carbon on the right, so we name it from the right, it is pent 2 in. The third molecule, the double bond is closer to the left, so the second carbon is again pent 2 in. So we can't have the same name twice. Therefore, we assume that pentene will only form 2 principal isomers, namely pent 1 in and pent 2 in, regardless of whether the double bond is on the right hand side or the left hand side. So long as it's on the second carbon, it is pent 2 in. And these are known as positional isomers. Therefore, we can assume and say that positional isomers differ in respect to position of the functional group. They differ in respect to position of the functional group. And therefore, in conclusion, we have said that isomerism is the existence of different structures of the same molecule. Thank you.